The first model in the reproduction goal is male reproduction. We will be discussing the structure and function of male anatomy as well as fertility requirements and sperm production. The objectives of this module are to identify the male reproductive structures and describe the function of each. You should be able to trace the pathway of sperm from synthesis to ejaculation by the end of this presentation, describe sperm production, and explain male fertility requirements. The primary sex organs in the male or the female are where the gametes, eggs or sperm, are made. They are also called gonads. In males, these are the testes. All other organs are considered secondary sex organs or accessory organs. Sometimes these are separated into organs that secrete fluids, like the glands, and the perineal organs, like the scrotum or the penis. Men have two testes, which are located in the scrotum. They descend before birth through the inguinal canal due to testosterone released during a key moment in development. This process is guided by a gubernaculum and becomes part of the spermatic cord. Originally, the testes are located in the posterior abdominal wall, and during embryonic development they descend down to the abdomen, although the inguinal canal, through the inguinal canal to reach the scrotum. They carry their neurovascular and lymphatic supply with them. If there's a failure to descend, no sperm are viable because it's too hot in the body. Ironically or weirdly, commonly the left testicle lies lower than the right. The inguinal canals are supposed to stay closed, but because the cords are there, it may weaken and lead to an inguinal hernia, which is actually quite common in men. The testes are located within the scrotum, and with the epididymis are situated on the posterior lateral aspect of each testicle. They are suspended from the abdomen by the spermatic cord, a collection of vessels, nerves, and ducts that supply nourishment to the testes. The seminiferous tubules are coils of tubes in the scrotum where the sperm develop, a half-mile of total distance. They are lined with epithelial cells called nurse cells or Sertoli cells, and their main function is to support the spermatogenic cells, which of course make sperm. There's also interstitial cells, also called Leydig cells, in the testes between the seminiferous tubules. These cells produce testosterone. The epididymis is the tube that leads out of the testes. The seminiferous tubules collect into it and carry the sperm to the next tube, which is referred to as the vas deferens. So the epididymis here in this picture is green and the vas deferens is blue. The process of making sperm is called spermatogenesis. Instead of mitosis, it is a process using meiosis. Meiosis is the process of cell division where you end up with haploid cells instead of mitosis, which you get identical diploid cells. So each sperm is going to have half of the dad's DNA compared to the original cell. Spermatogenesis is a 5-step process. The more mature they are, the more they look like sperm, and the closer to the epididymis they get to. Spermatogenesis occurs continuously from puberty till death and takes about 65 to 100 days, and it goes through these 5 steps: spermatogonia to primary spermatocytes, secondary spermatocytes, spermatotids, and then sperm. You can see this on the next diagram here. So upon sexual maturity, so at puberty, the spermatogonia cells are induced to start to replicate. Some of these become primary spermatocytes and go through the process of meiosis 1 to become 2 secondary spermatocytes. Others are made up here, are additional spermatogonia, which will be induced to do this later. So you have a continuous supply of spermatogonia that are going to be induced to do this process again until the man dies. Meiosis 2, you get from secondary spermatocytes, you get each of them produce 2 spermatotids. So now you have 4 spermatotids. They have half the DNA at this point, and they mature into what looks like our typical understanding of what sperm are. Okay, so each sperm ends up with half the DNA of the original primary spermatocyte. If you want more information or want to see this process in action, click on the link here. We'll talk about this in class because it's a little bit easier to see with a demonstration. But each parent cell has a pair of homologous chromosomes, and right before meiosis starts they double. And so in other words, the maternal and the paternal chromosomes replicate, and sometimes during that replication they trade things. And you can see the orange and the red overlap here. They separate, unattach, and then reattach, and when they get reattached, part of the chromosome that was once maternal is now attached to the paternal copy. What this ends up doing, if you look in this picture here, the crossing over ends up, so each sperm ends up with a slightly different copy of the genetic material, the DNA. It ensures that no two eggs and no two sperm are ever going to be identical, because we have 23 pairs of these chromosomes, so it's just an infinite number of different possibilities of chromosome mixtures that can happen. So every sperm is different. This is why when you have two kids 2 years apart, even if they're both boys or both girls, they are not identical children, because you're never going to have the same sperm meet the same egg twice in a lifetime. If you want a little update or a little review about meiosis, this is a great little wrap picture there, or link there. Okay, sperm are shaped like tadpoles, and they are the smallest cell in the human body if you don't count the flagella. They are the only cell in the human body that have flagella. The head contains just DNA, there's no cytoplasm, and is covered with a coating called the acrosome that has some enzymes that's going to help get into the egg when it meets the egg. There are mitochondria at the base of the flagella, and when they're mixed with semen, the sugar in it will power those mitochondria so the flagella can actually propel the sperm and move them. Here's a good picture. Mature sperm has flagella attached to pretty much just a nucleus. The head of the sperm contains only the DNA and a cap called the acrosome on top of it. It's going to help break down the shell, or the zona pellucida, of the egg. Alright, some of the rest of the parts of the male reproductive system. The epididymis is the tube from the seminiferous tubules to the vas deferens, or ductus deferens. It's about 7 meters in length but tightly coiled, and the sperm mature to motile little tadpoles here. Next, the vas deferens, or ductus deferens, is the tube from the testes to the prostate gland. It passes through the inguinal canal behind the bladder and then to the merges with the urethra in the prostate gland via the ejaculatory duct. The seminal vesicle is a sac or a gland which produces fluid that is alkaline, and it is going to release that fluid right ahead of the sperm because it needs to neutralize the urethra, which is acidic due to urine. The seminal vesicle secretion also contains fructose, a sugar for the sperm to use as energy, and it again is secreted into the ejaculatory duct right ahead of the sperm. It contributes to the formation of semen, and at this point the sperm now are going to be motile. The two tubes, the seminal vesicle opening and the vas deferens, merge in the ejaculatory duct, which is in the prostate gland. The prostate gland also secretes a fluid ahead of the sperm into the urethra. It too is neutralizing, but it also contains some other things in there, antibiotic even, to help prevent UTIs, as well as other chemicals that help stabilize the sperm. The gland itself is about the size of a chestnut, and because it surrounds the urethra, inflammation or swelling of it can impede urination. So it's not only affects the reproductive system when you have prostate problems, but also the urinary system. The bulbourethral gland is a small gland that secretes a fluid that helps lubricate the end of the penis. Its secretion is also dumped into the urethra, again ahead of the sperm. The semen, therefore, is an alkaline fluid that is both nutritious and protective for sperm. It allows for capacitation. It's going to help weaken the acrosome so those enzymes can be released when they get up to the egg. It also makes the sperm motile. So until sperm is mixed with all these solutions, it does not move. Again, the sperm don't have to swim out of a guy, they're shot out of the guy during ejaculation. They only need to be motile once they're in the female. In this diagram you can see the testes inside the scrotum, the epididymis, the vas deferens, the prostate gland and seminal vesicle, and then the urethra and the bulbourethral gland. Okay, the next couple of slides are going to talk about the external genitalia. The scrotum is a pouch of skin holding the testes. It hangs outside the body because sperm like it a little bit cooler than body temperature. In fact, if the testes are held up too close to the body wall, the man will be infertile because the sperm will fry and get too hot. The scrotum is very similar in structure to the female labia. The penis carries both urine and semen through the urethra to the outside of the body. The body of the penis contains tissue called the corpus cavernosum and the corpus spongiosum. The spongiosum is the one closer to the urethra. It's in red here. Both of them allow for erection. As blood flows in during the erection process, it can't flow out, and it allows for an erection to happen because of the vasodilation. At the end of the penis is called the glans penis. That's where all the nerve endings are. It's also comparable to the clitoris in the female. In fact, transgender women who want to become men, the clitoris can be used sometimes surgically to make them a small penis. The covering of the penis is sometimes called the foreskin and is sometimes removed if the baby is circumcised when born. During sexual stimulation, the penile arteries dilate due to nitric oxide, and more blood flows in than flows out, allowing for or resulting in an erection. Once orgasm is reached and ejaculation occurs, the parasympathetic nervous system becomes less active and the sympathetic nervous system is now active. That reduces the vasodilation and the blood can flow out and the erection is lost. Again, fluid from the bulbourethral gland is released first, then the prostate, then the seminal vesicles, and behind that the sperm. So again, erection is a result of the parasympathetic nervous system releasing nitric oxide. Blood flows in and accumulates, and it can't leave as fast as it goes in, allows for a reaction, an erection. Viagra stimulates this. Okay. Emission or orgasm occurs when a man ejaculates. This causes sperm to be pushed into the urethra. Semen is made, mixing occurs, and because of the act of ejaculation requires sympathetic nervous system activation, it then slows the vasodilation and the erection is lost. So parasympathetic first to get an erection, and then sympathetic nervous system to get ejaculation. When it comes to men and fertility, the first thing a doctor might ask is, are you able to get an erection? And are you able to get to ejaculation? Both of these must occur. In other words, a man has to reach orgasm in order for him to be fertile. Sometimes different drugs that the person is on, or stress, or other medical problems can prevent erection or ejaculation. And if those can be fixed, fertility sometimes can be fixed. So again, male fertility, one of the things they'll ask is erection and ejaculation, is it working? Then they'll look at volume of semen. Good semen has 120 million sperm per mil, and 2 to 3 mils is normal. Less than 100 million per mil is considered somewhat infertile, and under 20 million per mil is considered functionally sterile. So they want the volume and the number to be high enough for fertility to occur. Other things they will look at is, do the sperm swim? Are they viable? Are the flagella working? Is the shape normal? It has been shown that abnormally shaped sperm, or sperm that don't aren't as motile as others may have DNA damage, and you wouldn't want them to fertilize an egg. Some toxins have been known to increase infertility. Some of the chemicals used in dry cleaning. Bicycle riders without the right kind of equipment or protection will sometimes have lower fertility rates because the sperm get too hot. The amount of testosterone is regulated by negative feedback primarily. It's constantly being produced, and while it may decrease as a man gets older, there's always going to be some. When men are partnered in long-term relationships or become parents, testosterone declines, and the more a man spends with his offspring, the greater the decline. The theory is that there's less need to reproduce once he has children, which is kind of weird. Testosterone may reduce with age, but American men have a bigger decline than other countries. This study was done in 2002, and I always find it very interesting that the top line is American men and the bottom green line is South American. The blue line is from Nepal, so Asian, and the red line is African men. So there's a lot of difference at the beginning, but by the end everybody's down to about the same amount. I have no idea why. At puberty and beyond, testosterone contributes to bone density, fat distribution, muscle mass, muscle strength, red blood cell production, sex drive, and sperm production. Men have more red blood cells because it stimulates erythropoietin production. Men have more muscle because it increases muscle growth and development, particularly in conjunction with growth hormone. So testosterone plays a lot of roles in a lot of different areas of the body. Once puberty hits, the differences in testosterone and estrogen are striking in boys and girls. But before that, like at age 10, if you actually measured testosterone and estrogen levels in kids, there would be no difference. You wouldn't be able to tell which one was a boy or a girl based just on that alone. Hopefully you remember this from the endocrine chapter. Again, testosterone levels are regulated by the hypothalamus pituitary pathway and negative feedback, so high levels of testosterone, lower LH and GnRH, which then not as much testosterone would be released. So testosterone levels stay fairly constant. There's not nearly the cyclical things that occur in women when it comes to estrogen and progesterone. Men, testosterone levels tend to stay fairly constant. Another hormone in this picture we haven't talked about before is inhibin. It is also secreted from the testes, from the nurse cells, and it too can use the negative feedback system to lower FSH. In fact, it could be used if somebody gave you extra inhibin, gave a guy extra inhibin, it would lower FSH to the point that it would block spermatogenesis. So it could be used as a birth control pill. We've covered infertility, and again, about half of the infertility problems between couples are related to the male and the other half related to the female, although a portion of both are unknown or unexplainable. Sperm count, morphology, and motility, as well as erection and ejaculation problems, are all reasons for infertility. Sexually transmitted diseases or sexually transmitted infections, sometimes called STIs now, are often without symptoms in men, but men can still pass them to their partners. It can lead to problems in men if untreated. HPV, which causes genital warts, also increases the man's risk of penile and anal cancers. The lifetime odds of acquiring HPV is 91.3% in men, so getting the vaccine is probably a good idea. Prostate cancer is the most common cancer in men. Almost always, if you live long enough, you'll probably have some swelling or something with your prostate, although you may die of something else sooner than that. There's a variety of treatments for prostate cancer. Besides surgery, they do radiation as well as some other medication-like type things. If you need some more information, there's information in your textbook, online, in the lesson. Be really careful what you Google because you can get some pretty crazy stuff.